Cell is called a structural and functional unit of life, of all living organism capable of independent existence and can perform all function of life. First, we will go through prokaryotic cell structure. It is a single cell organism. It includes membrane of two-domain, archaea, and bacteria. Let's observe structure of bacteria. Let's start with surface protection of bacteria. Cell envelope have three-layer structure. First is outer layer. This is simple layer which is likely loose sheet or encapsule tough structure. Outer layer is made up of glycocalyx. Basically, polysaccharide on external surface is called glycocalyx. Glycocalyx is a layer of carbohydrates. The carbohydrate chains attached to proteins in the case of glycoproteins or lipid in the case of glycolipids. Organized polysaccharide network that is present within a capsule. Example of capsule bacteria are E. coli and Klebsiella pneumonia. The main function of bacterial capsule is to provide protection to the bacteria. This protective nature of the capsule interferes with the host immune response, allowing the bacterium to evade the body defenses and increase its chances of causing infection by making it more challenging for certain type of WBCS to engulf and destroy the bacteria through phagocytosis. Unorganized, loosely attached polysaccharide layer present in the slime layer bacteria. This slimy or mucoid layer can aid in the establishment of infection by allowing bacteria to adhere more effectively to surfaces, including host cells. Example of slime layer. Bacteria are Streptococcus mutant and Pseudomonas bacteria. The outer membrane of bacteria is typically a distinct structure and is part of the bacterial cell envelope. It is located outside the cell wall. The outer membrane contains various components, including lipopolysaccharides, LPS, porins, and proteins. Porins, they are the little proteins that allow for the transport of certain type of molecule or substance in and out of the bacterial cell wall. Lipopolysaccharides, LPS, as endotoxin, LPS is indeed considered an endotoxin, and its lipid A component is responsible for its endotoxic properties. Lipid A has the ability to stimulate immune system cells to release massive amounts of cytokines, contributing to the inflammatory response. Core polysaccharide. The core polysaccharide is an intermediate region between lipid A and the O antigen in the LPS structure. While it contributes to the stability of the LPS molecule, it is not directly involved in the immune stimulation as much as the lipid A component. And the O antigen, the outermost part of LPS, is indeed involved in immune recognition and antibody attachment. This is the general role of LPS components in interacting with the immune system and their contribution to bacterial pathogenicity. The outer membrane only present in gram-negative bacteria. Middle layer is called cell wall. The cell wall layer composition is vary between different kind of cell. It's typically made up of peptidoglycan. In the bacterial cell wall, N-acetylglucosamine, NAG, and N-acetylmuramic acid, NAM, are polymerized to form the glycan backbone of peptidoglycan. This backbone is then cross-linked by short peptide chains, creating a mesh-like structure. That provides strength to the cell wall. This structure is essential for maintaining cell shape and integrity. The arrangement of the peptidoglycan determines the shape of the bacterium. Different bacteria have distinct cell wall structures. Gram-positive bacteria have a thick peptidoglycan layer. This layer is located on the outer side of the cytoplasmic membrane. This layer contributes 90% of the cell wall's weight. On the other hand, the gram-negative bacteria have a thinner peptidoglycan layer. This layer is sandwiched between the inner cytoplasmic membrane and the outer membrane. This layer accounts for a smaller percentage of the cell wall in gram-negative bacteria. The periplasmic space is a clearly defined gel field region. Between the inner and outer membrane in gram-negative bacteria, it is a specific space. That is not present in the same way in gram-positive bacteria because it lack the outer membrane. This space is significant for various function by includes variety of enzymes and proteins. First, nutrient-binding proteins. It help for the uptake of essential nutrients from the external environment. Second, detoxifying enzymes are give help to bacteria for deal with harmful substances. Third, periplasmic chaperones which assist in the folding and stabilization of other protein in periplasm. And last one is beta-lactamase enzyme which play a crucial role in antibiotic resistance like penicillin. The last layer of cell envelope in bacterial structure is plasma membrane or cell membrane. Plasma membrane is primarily composed of a phospholipid bilayer, a molecule with hydrophilic head and hydrophobic tail. Protein and cholesterol are also included in the composition of plasma membrane. This bilayer is integrated with protein molecules distributed throughout the layer. Some proteins are found to the external part of the layer. This is called peripheral protein, and some proteins are totally merged into the layer and are called integral protein. Integral proteins are strongly bound. They can only be separated when membrane is destructed. Peripheral proteins are loosely attached. They can relocate their position within a membrane. The cholesterol molecule present in the membrane provides stability to bilayer. They help the bilayer not to get separated too much in different environment conditions. Arrangement of these molecules form a barrier that separate the interior of the cell from its external environment. Control the entry and exits of selective biomolecule. The protein play crucial role in various cellular function, may involved in transport processes, cell signaling, and maintain the structure integrity of the membrane. We will discuss some other surface protection of bacteria in next video.